Good day everyone. Today we are discussing artificial seeds. So artificial seed is defined as artificial encapsulation of somatic embryos, shoot bud or aggregate of any cells or tissues which has the ability to develop into a new plant in vitro or ex vivo. So yesterday we discussed about somatic embryos. These somatic embryos can be encapsulated. Artificial seeds are also called as synthetic seeds. So we are going to produce a man-made or a artificial seed from the somatic embryos that we have prepared. Okay, let us see how it is done. So we're coming to the history of it. Artificial seed was first introduced in 1970s, and the production of artificial seeds is used, which is used for plants which do not produce viable seeds. Okay, artificial seeds are very small size. They are advantageous because they can be stored for a longer period of time. They are easy to handle and they can be shipped. Okay, embling is also used for the origination of synthetic seeds. The use of synthetic varieties for commercial cultivation was first suggested in maize by Hayes and Garber in 1919. So, those plants which do not produce viable seeds, then these artificial seeds can be used. So, based on the techniques, two types of artificial seeds are produced. One is desiccated synthetic seeds. Here, they are produced naked, that is, they are not encapsulated in any medium. Next is hydrated synthetic seeds. They are produced by encapsulating the somatic embryos in some substances like hydrogels, like sodium alginate, potassium alginate, carrageenan, sodium pectate or sodium alginate with gelatin. So two types of artificial seeds are desiccated synthetic seeds and hydrated synthetic seeds. So desiccated synthetic seeds means they are in the dried form, whereas hydrated synthetic seeds means these seeds or somatic embryos are encapsulated in some gel-like substance. So the need for artificial production, here the development of micropropagation technique will ensure that abundant supply of desired plant species are available. Then the development of artificial seed production technology is currently considered an effective and efficient method for the propagation in several commercially important agronomic and horticultural crops. Artificial seed also be a channel for new plant lines and also advantages to be delivered directly to greenhouse and field. Then high volume of propagation, that is, by clonal propagation we can get the same like individual similar to that of the parent, and also for commercially important crop species also we can produce many high amount of propagules by using this artificial seed production. So what are the what is the need for it? One is by abundant supply of desired plant species can be brought about. Then the plants which have agronomic and horticultural crops can be commercially, they can be produced. Then these seeds can be they can be transferred to greenhouse or fields. And the clonal propagation, by clonal propagation, a high amount of or high volume of somatic embryos can be prepared, or they can be encapsulated, and they can be commercially used. They can be used for commercially to propagate these plants. So this is the need for why we are using this synthetic seeds. Okay, so what are the basic requirements for the production of artificial seed? One is we have to have vigorous somatic embryos that can produce plants with maximum frequency. Then it should be inexpensive production. So large number of high quality somatic embryos are produced in an inexpensive manner. Then encapsulation and coating system, it is not a limiting factor because somatic embryos can be encapsulated or coated coated and it is very easy to produce also. Then the commercialization of these synthetic seeds are also possible. So these are the pre-requirements. First we should have vigorous somatic embryos that can produce good plants. Then it should be inexpensive production of somatic embryos. High quality somatic embryos should be possible. Then encapsulation and coating system should be there, which should not be a limiting factor. Then there should be any medium for commercialization of these synthetic seeds. Now let's see how these synthetic seeds are produced. First thing, we have to establish the somatic embryos. Then these somatic embryos should be mature somatic embryos. They should be synchronized. That means should be all of the same age group. Then mass production of somatic embryos should be produced. Then the encapsulation, that should be standardized. Then artificial endosperm should also be provided. Then like this, with all these requirements, mass production of artificial seeds are produced, and then they can be transferred to greenhouse or field directly. These artificial seeds can be directly transferred to greenhouse or field. So what are the requirements? One, we should have somatic embryos that should be established. Then another thing is the encapsulation should be standardized. Then artificial endosperm should also be provided, that also should be standardized, and then they can be directly transferred to the greenhouse or field. So once we have done that, we'll see how these synthetic seeds will look like. So these are the final product. This is how a synthetic seed will be seen. Now we can see that the somatic embryos are encapsulated in jelly like substance. Okay, these are synthetic seeds. They are artificially made seeds. This can be directly transferred to the field or to the greenhouse. So let's see how these artificial seeds are encapsulated. We have already studied how artificial seeds or somatic embryos are made. Now we are seeing how they can be encapsulated. These somatic embryos are dipped in hydrogel. The hydrogel may be anything, that is alginate, that is sodium alginate, or agar, or sodium alginate solution, which is prepared in MS basal medium, or somatic embryos that are dipped in such solution like hydrogels, and they are coated by placing in a flask kept on a magnetic stirrer. So for 20 to 30 minutes we keep it in a magnetic stirrer. The embryos are put in hydrogels, which these hydrogels will be coated on these seeds. Okay, the hydrogels will be coated on the somatic embryos, and that's how we produce the artificial seeds. So there are many methods for it. We'll see how what are the different methods also. Now when you take certain gels like sodium alginate, there is a specific proportion, like 1 to 5% of sodium alginate solution should be prepared. And so let us see how the whole process will be done. The embryos will get covered by the calcium alginate, which is a stable complex because it forms a ionic bond between the somatic embryo. Then these gelled embryos, they are washed in water or with basal medium, and the synthetic seeds are ready for use. Okay, so the somatic embryos, by keeping in a rotary shaker, the calcium alginate will be formed around it. Another type of or another method is dropping procedure. Here the somatic embryos are put into or dipped in 2 to 3% of sodium alginate, and they are made to fall as a drop from the funnel. Okay, so these somatic embryos are kept in the calcium salt solution for 20 minutes, and the calcium alginate forms a gel, or it forms a gel like on top of these embryos. Then it can be rinsed in water and can be stored in airtight container. Another method is molding method. Here the embryos are mixed with a temperature dependent gel like Gelrite or agar, and the cells get coated with this gel at lowering of the temperature. So as it cools down, the gel get coated on it. Now let's see how artificial endosperm is provided. Somatic embryos, we know they lack seed coat or the testa, so and also the endosperm, and therefore they should be provided with artificial endosperm for protection and nutrition of the zygotic embryos. So in order to augment these deficiencies, nutrient and growth regulators are added to the encapsulation medium. Okay, this results in increased efficiency of germination and also for the viability of encapsulated somatic embryos. These synthetic seeds can be stored for longer period of time, even up to 6 months, without losing its viability, and it should be stored at 40 degrees Celsius. These seeds should be stored at 40 degrees Celsius. So let's see what are the adjuvants to be added to the medium. So one is to prevent the embryo from desiccation and mechanical injury. Many materials like nutrients, fungicides, antibiotics, then certain microorganisms like rhizobia, all this can be incorporated into the encapsulation medium. So even activated charcoal can be provided. Activated charcoal improves the conversion and vigor of the encapsulated somatic embryos, and it retains the nutrients within the hydrogel capsule, and it slowly lets them to release into the growing embryo. So these are the adjuvants. That means apart from the nutrients or growth promoters or hormones that we are already providing, even these can be added, that is fungicides, pesticides, antibiotics, or rhizobia like microorganisms or activated charcoal. These are supplements that can be added to the somatic embryos in order to retain their nutrients and also provide the nutrients in a slowly to the growing embryo, so that these somatic embryos or these artificial seeds have greater viability or better viability, and they can be stored for a longer period of time. Now next thing is the potential use of artificial seeds. We have already discussed this earlier, because we can transfer it to greenhouses and field. Then the cost of production of the seeds are very less. Then elite, that is new plants with elite characters can be produced. Selective genotypes can be produced. Then large scale monoculture, we have seen that in tissue culture we get clone like individuals. So monocultures can be produced in large scale. Then by even adding adjuvants like microorganisms or pesticides or fungicides, no need for extra effort for adding all this. Then they can be conveniently handled because they are very easy to be transported. Then they can be produced throughout the years. We know some plants they produce seeds only in a season, but somatic, if we can use somatic embryos, then we are able to get seeds throughout the year. Then the germplasm conservation is possible. Then large scale production of identical embryos in a short period of time is possible. Then we have already discussed that this synthetic seed production is used for those plants which do not produce viable seeds, isn't it? So the plants which do not produce viable seeds, those plants can be easily propagated by these artificial seeds. So I hope you understood this section. If you have any doubts, please be free to ask.